Hello, everyone, and welcome to the Mechanochemistry discussions. This seminar series is hosted by the NSF-funded Center for Mechanical Control of Chemistry, or the CMCC. I'm Ashley Martini. As always, the goal of the center is to bring the mechanochemistry community together. Our seminars stream live on the third Thursday of every month at 11 a.m. Eastern and are available to watch anytime on the YouTube channel. Over the past few years, we've had a great group of speakers, and again, they are all available to watch anytime on the YouTube channel. We also have an excellent group of speakers planned for the upcoming months, and we hope that you will join us for all of them. Before we get started, a few thank yous. First, Dr. James Batiste, the Center Director, Jennifer Belsick, the Center's Administrative Coordinator, and Katie Floyd and Ryan Kwok, CMCC students, without whom the seminar series would not be possible. Thank you so much for joining us. Please do subscribe to the YouTube channel and see all of our excellent seminars. Last thing, just a reminder that the webinar is being recorded. If you have questions, you can either email them to us at cmccdiscussions@gmail.com, or you can post them directly into the YouTube channel. Either way, they will be propagated to the speaker at the end of this seminar. Last but not least, it's my great pleasure to introduce today's speaker, Professor Chelsea Davis. Professor Davis joined the faculty of the University of Delaware as an associate professor in the departments of mechanical engineering and material science and engineering in 2023. Previously, she worked in the School of Materials Engineering at Purdue University, where she established the Illuminating Interfacial Mechanics Lab, focusing on the development of micromechanical characterization tools to investigate the interfacial and surface properties of soft materials. Dr. Davis obtained a bachelor's in textile engineering from North Carolina State University and a master's and PhD in polymer science and engineering from the University of Massachusetts Amherst. Dr. Davis was a Michelin postdoctoral fellow at the ESPCI in Paris, and then an NRC postdoctoral fellow in the Polymer and Complex Fluids Group at the National Institute of Standards. Dr. Davis's work has been recognized through the NSF Career Award and the Adhesion Society Early Career Scientist Award. Today is my pleasure to welcome Dr. Davis to the Mechanochemistry discussions.
All right, thank you, Dr. Martini, for that introduction, and welcome everyone to this CMCC YouTube seminar. As she said, my name is Professor Chelsea Davis, and I'm an associate professor at the Department of Mechanical Engineering and the Department of Materials Science and Engineering at the University of Delaware. Today, I'm excited to share with you some of the work that we've been doing over the past 6 or 7 years, where we're trying to utilize mechanophores, these mechano-responsive materials I'll introduce you to in a moment, in order to quantify stresses within polymer composites. So although I fully recognize this is a mechanochemistry seminar, today we're going to be focusing quite a bit on utilizing these mechanophores, targeting more engineering applications within composite materials. So with that, I'll go ahead and get started and just let you know that I've organized this talk into 2 sections. The first section, I want to share with you some of the work that we've been doing to try to quantify the fluorescence response of a fluorescently responsive mechanophore within these composite materials, and then how we've taken that quantified stress mapping capability that we now have in order to investigate some classical composite mechanics questions and problems in order to visualize how stresses concentrate in loaded materials. In the last couple of slides, I'm going to show you some very new work that we are on the cusp of submitting that is focusing more on sort of the mechanics or deformation of bulk elastomers. So this isn't necessarily composites, it isn't composites, but it is still exploring how mechanophores can elucidate further phenomena within fracture and cutting mechanics of elastomers. So with that, we're going to go ahead and get started and focus on these composite systems. So for some of you that are coming from the chemistry world, you might not be as familiar with fiber reinforced or rigid particle reinforced polymer matrix composites. I know all of us use composites in our daily lives. We've all seen fiberglass and carbon fiber composites that are being used for bike frames and tennis rackets and airplanes and that sort of thing. But what we care about as mechanical engineers and also as composites engineers is how can we prevent these materials from failing upon loading? And the failure mechanism for fiber reinforced or particle reinforced polymer composites can be really unique and different from how metals fail or how ceramics fail because there are so many interfaces within these materials that there are a lot more opportunities for defects to arise at the interface that then propagate and can lead to some really catastrophic failure events. So what we've been focusing on in my group is how can we use these mechano-responsive materials to highlight where stress is concentrating and then to observe how, if failure initiates, how does it propagate through the material? What is the chain of events of stresses that get distributed through the material as it fails? So what we're going to be looking at today is how stresses accumulate and then lead to failure around round and how should I say this, flat fiber end geometries. So we're going to be looking right at the fiber tip of this chopped fiberglass you would see in all sorts of sort of moldable composite systems. So as I said, we're going to have two main goals of this particular portion of our research. We're going to be trying to visualize these localized stresses and develop a quantitative strategy that allows us to take pictures of the mechanophore that's activating and interpret that or interpret that as stress distributions within our material. So we did not invent mechanophores within my group. We are not chemists, and we are standing on the shoulders of giants in the sense of we are utilizing mechanophores that have already been developed in the labs of Jeff Moore and Nancy Sotos, as well as we're using the particular form of spiropyran that has come out of the lab of Professor Steve Craig at Duke. And these mechanophores have been shown over the past couple of decades to have this really, really specific change in their color or change in their intensity as a function of how much mechanical deformation you apply to the system. So when we first started working on mechanophores in my lab, I kept getting the question of if you pull on it, does it get brighter? And in that, you have to be really specific about what it is. If you're talking about it, the continuum system that you're pulling on, yeah, the more you pull on this object, the more color saturation you'll observe, or the brighter, the higher the fluorescence intensity you'll observe. But if you're talking about an individual molecule, these are fluorescent molecules that have a very specific quantum efficiency or a very specific brightness. You can think of them as a binary reporter, right? They are on or they are off, and that's it. So what's really happening when we pull on one of these highly deformable elastomeric polymer networks is we're activating new populations of mechanophores with every subsequent force here or every subsequent strain step that we're taking. We're turning on different populations of mechanophores as we elongate and stretch the longer chains in our network. So if we think about how this correlation of number of mechanophores activated to amount of deformation in our material correlates, we can then see that we can start to develop some fairly straightforward relationship between how bright our sample looks to how much deformation is in the material. And we wrote a review article a couple of years back trying to summarize some of this work in an engineer's introduction to mechanophores. And in that review article, we talked about sort of the two classes of mechanophores that are most popular in the literature right now. And these all stem from some sort of bond scission event that occurs when you apply a force across a labile bond And by biasing sort of the activation energy or the energy required to pop that bond, you can induce an isomerization in the case of some of these reversible mechanophores, like the spirolactam I'm showing here, or you can induce a selective scission event where the molecule becomes two molecules that will either both fluoresce or one half of the molecule will fluoresce after the scission event. And these tend to be irreversible mechanophores. So the particular molecule that we're using in my group right now, our workhorse is spiropyran, particularly the nitro form of spiropyran. And when we have this molecule in the unactivated state, you can see this carbon-oxygen bond is going to be the bond that we're targeting. So we're applying a load at these two polymer handles, and if we apply sufficient force, you'll observe that the molecule opens up, goes to an isomerated state, or an isomer of itself, and you can see the conjugated state that it adopts, and this is where the fluorescent center of this molecule is going to be found. So this particular molecule is really fun because if we put it into a transparent silicone, which is most of what I'll be showing you today, we can write the Davis Research Group's initials and see them under a fluorescence microscope. And we also get this visual color change that requires just our eyeballs to be able to identify. So with all of this, this is really nice, but so far everything I've shown you is on or off, more or less. It's really, really qualitative and sort of wishy-washy. And what we wanted to be able to do was to put numbers. Can we say it is this bright, therefore the stress is X at this location within our material? And we weren't the first people to think about this. So several groups have used either differential image correlation. This is where you put a bunch of speckles onto your system and then track how those speckles move relative to each other in order to map a strain field onto your material. Or several other groups have utilized finite element modeling in order to say, to calculate what the stresses should be within their material. And two of the first works that came out trying to correlate this stress response to the fluorescence intensity of mechanophores were looking at the stress concentration ahead of a propagating crack. So if you take something like a multiple network gel and you put a notch in it and then deform the material, as you deform the material further and further, you get a larger stress field arising around the tip of that crack. And they correlated this to the activation or the intensity of their mechanophore. And this has happened in rubbery and glassy systems. But for us, we're working with polydimethylsiloxane. We're working with Dow-Silgard 184, sort of the classical perfect elastomer, which is a terrible way to describe it, but it has become a material that a lot of people in the soft matter mechanics of materials community use as sort of the test subject of choice. That material, while you can extend it quite significantly, is also really brittle if you get a notch in it. So it can fail brittly, and the crack will propagate in an unstable way once you notch that material, making it really hard to keep looking at the crack tip. So my group, when my first PhD student joined the lab, we thought, well, is there another way to concentrate stress within a composite material? So what Mitch Renchek did when he first joined the lab was rather than focusing at the tip of a propagating crack, he chose to focus on the poles of a rigid inclusion. So this is a little glass bead that we've put inside of a big chunk of this transparent mechanophore functionalized silicone. And if we deform this single particle composite, we can observe at the poles a really significant stress concentration and stress distribution occurring near the surface of our sphere. If we then take this, which is maybe one of the easiest geometries possible to simulate with finite element analysis, we can calculate what the stress field should look like in one of these loaded samples. And then by simply applying a spatial correlation between what we've calculated the stress should be and what the fluorescence intensity is that we observe in our microscope, we're able to get some sort of quasi-linear relationship between the mechanophore activation and the deformation of our material. And this was the first mechanophore paper that came out of my lab several years ago, showing how we had performed this analysis. So in order to do this, I do want to tell you a little bit more about the system we're using, the mechanics of the experiment. So all that we're doing is we're combining a micromechanical load frame that is going to deform the sample laterally over the objective of a laser scanning confocal microscope. So this is what it looks like as a straightforward cartoon, where we have the bead centered over this big circle here as our objective. This is what it looks like when you look down at the microscope, where the particle is about 1 millimeter across, and we can see the objective underneath it. And then here is just just an image of the Silotech Mu-TS that we utilize in order to perform these experiments. So there was quite a bit of materials characterization and sort of process refinement leading up to being able to do this. And after Mitch graduated, another graduate student in my lab, Jared Gohl, came in and really helped us with the chemistry to refine and control the interface between our particle and matrix. So as I said at the beginning, what we care about is composite interfaces. So what Jared did with his undergrad research assistant, Tristan Wiley, is they functionalized the glass particle before embedding it into our Mechanophore functionalized silicone with either a long perfluorinated silane, a short perfluorinated silane, or a vinyl termination. And this was our attempt to get low, medium, and high adhesion, because in the case of the vinyl terminated silane, we could induce covalent attachment as we cured our PDMS matrix around the particle. And this was really successful. This was a fairly straightforward strategy that allowed us, if we look as a function of strain, I'm pulling up and down on this object. And here in the low adhesion case, the sphere acts as sort of a void in our sample because there's no real crosstalk or interaction across the interface. And you can see the mechanophore starts to light up at the poles where the particle is preventing the hole from adopting the full ellipsoidal grain of rice void structure that it wants. But what's more interesting for us was we observed really interesting gent-park debonding or gent-park cavitation that occurs when you have a weak-ish interface that holds for a while, stores a lot of mechanical energy, and then leads to a large-scale cavitation event. So this you can think of as like a hollow Easter egg that has opened up at the top and bottom of our particle. And I don't know if I can play these again. Let me try. Um, for the high adhesion case, what we were really going for, this was not the highest adhesion case. We improved our method a little bit beyond that. But if we keep going, we're already at 40, 45, 50% strain. And you can see the interface is starting to fail in the two lower cases. But here, what isn't it going? There we go. You can start to see stress happening around 90, 100% strain. And we're going to get a gent-park debonding on one side, but we really care about this right here. This is going to allow us to perform the calibration that I showed you on the previous slide. So if we see this as a matrix, this was really just in case the videos didn't work. We can then take this and show that we have really good agreement between our simulation and what we observe in our fluorescence microscope. So from there, we perform our mapping where we take our fluorescence intensity, we take our finite element stress intensity calculations, and we're able to develop a false color lookup table that we can apply where we have colored our fluorescence intensity as a function of stress, or turned it into stress. And for this particular set of experiments, we found a correlation coefficient of about 0.23, which allowed us to create this color map. So now we've developed this really exciting tool that will allow us to perform this calibration. So what I'm showing you is just what I showed on the previous slide now in video format during one of these experiments where we have the fluorescence intensity, we have the FEA doing absolutely nothing until about 70% strain, and then we have our calibrated image where we have mapped these two and you can see the strain. Okay, I am underwhelmed that this works because this is the experiment we calibrated to. But now we can take that same calibration, and if we take two particles that start at one radius of separation and start pulling them apart, what you'll observe is we get this really exciting uh, crosstalk or concentrated stress event where you have these two particles really close to each other. And this starts to become important in particle reinforced elastomers, like the tires that you drove to work on. And we can go further. We can start to look at stress concentrations around corners, which we've all studied in sophomore, well, what the mechanical engineers of us have studied in like our sophomore mechanics and solids courses, where corners or really sharp radii of curvature features tend to concentrate stresses. And that's what we observe if we put a sharp shard of fiberglass or glass fiber into our system, either orthogonal to the stretching direction or parallel to the stretching direction. In either case, around these sharp corners is where we see all of the stress concentrating. And I particularly love this image or this video because it really highlights why we're doing this. Why are we trying to use mechanophores in order to visualize stresses? And it's because it allows us to see features or stress concentrations on the order of tens of microns, which we haven't necessarily been able to do before. So now that we have this hammer, we kept looking for other nails. And the next student to join my group came back to this question and was like, well, we've seen what happens around a single spherical particle, but what happens at the end of a fiber if you start messing with the shape of that geometry? So in short fiber reinforced composites, some people are starting to propose modifications to the geometry of these fibers in order to toughen the whole system and prevent fracture events, or at least enhance their fracture toughness. So my current senior student in my lab, Nazmul Hawk, has been working on just that. We recently published an article where he took really big glass fibers and performed single fiber pullout experiments where he was still using his Mechanophore functionalized matrix. And now he's going to pull one giant fiber out of out of the out of the matrix, and he's messed around with the shape of these different fibers, going from round all the way to flat. And he was really careful to follow some of the analysis that already exists in literature from the 90s in order to ensure that all of his experiments, we had the bottom of the fiber, the failure event occurred at the bottom of the fiber or the embedded end rather than at the emergent end and then having the matrix just unzip from the top down. And he's performed all of this analysis to ensure that his geometry and modulus mismatch between fiber and matrix was such that we always get embedded fiber and separation. So his setup looks very similar to the setup I showed you before, only now instead of holding the whole sample, we're going to use one grip to hold the matrix and we're going to use the other grip to clamp the fiber. And we're still going to look right here at the fiber end. But for all of the experiments that I show you, in my head, it's just easier to think of the system as upside down. In reality, we look down at it, so it doesn't matter how you turn your head. But these are how we're going to be looking at these experiments. So this is a round fiber geometry. Um, and we can go through and characterize the load and displacement relationship of all of these. So for all of the different, or the 4 different sample geometries that we utilize, we see a fairly systematic difference in where the fiber end starts to fail or pull out of the matrix. And then we see 2 very distinct between the round and the sharp. We see some distinct thick slip sliding events But then for the cone and the flat, we saw much smoother curves, and we also saw separation at lower forces. So what those corresponded to for the flat oh, I've got my colors wrong. I did this wrong. This is the flat geometry. This should correspond to this orange case. I wanted to show you the extremes. So this is the flat geometry where you can see that we have very intense activation, but in a very specific way where we only are observing mechanophore activation due to sliding or the very high forces that are occurring right at the edge. And we only saw a tiny bit of activation right at the edge. Whereas if I play the round fiber geometry again for comparison, you can see that we get this very large region of activation prior to we actually get failure in the bulk that I cut out. I cut out that frame. So we can go through and utilize mechanophores in order to see stress concentrations around all of these different fibers as they're being pulled out and then pulled through the matrix. And we can go through because we have the ability to see really small features, or not really small in this case, but we can see small features and start to do a little bit more thorough analysis in order to understand how good of a job our calibration is doing at describing what we expect to see based on what the finite element calculations of the stresses in our matrix are. So here I'll show that at 45 and 90 degrees, Nazmol got really great agreement between his experimental intensity as a function of position. So you can think of this as how much stress is occurring in the system relative to the location or the shape of his system. However, for his 0 degree, or right here in the direction of the pulling, or directly opposite to the pulling, we observed a much more diffuse or larger deformation zone than the much more narrow, concentrated stress concentration that was observed or was calculated for the finite element analysis. And you can see that reflected here where this solid line is the FEA, and it's way higher than way higher and sort of tails off in a different way than this more diffuse observation that we had. And we believe that this is due to the fact that we're using a perfectly elastic mesh for our FEA, and we're not incorporating any damage or allowing any damage or chain scission events, which are almost certainly happening at these high loadings within our polymer network. So we're working to see if we can't incorporate the idea of damage and stress, stress redistribution as a function of damage within our network. So we were able to perform or to show a matrix. This is just a figure out of that article from Soft Matter earlier this year, where we observed just the different fracture events that occur around these different fiber geometries. So this is all work so far that has been done in the bulk. But my background has always been caring more about interfaces than bulk deformation. So I was always curious and have been wanting for a very long time to install mechanophores at the interface between a reinforcing particle and the polymer matrix, rather than putting the mechanophore in the bulk itself. And I had this opportunity when my group was awarded a small contract from the Semiconductor Research Corporation, where in microelectronic chips, there's a concern when you have the microelectronics that are sensitive to humidity and oxidation and all sorts of environmental degradation modes that are always encapsulated in some form of polymer matrix. And if the interface fails, you have to be able to detect that delamination or debonding event before you can see it, before it's big enough that you can see. And we thought mechanophores might be a really exciting way to interrogate molecular scale or sub-nanometer even separations in the interface. So in order to do this, you have to think about designing a mechanophore, and this is the challenge for the chemists on the call. You have to think about designing a mechanophore that has what my group calls an asymmetric functionality. You can't have the same handle on both sides of your mechanophore. You have to have some sort of different chemical reactive group or reactive species on the two sides of your mechanophore. If you don't, then whichever surface you functionalize first is going to end up with the mechanophore just attached in a loop across the surface. So if you're going to have a silicone-silicone interface, that's going to require a different functionality, or a mechanophore with very different functionality than one that you're going to attach between an epoxy silicon interface. And these are both encapsulating materials that are used in the semiconductor industry. So this is very much ongoing work. All that we've done so far is published a proof of concept. Others have started or have published several explorations of these, mostly looking at particles that are functionalized with a mechanophore that have some sort of polymer tether that entangles into the polymer network or entangles into the solution or a suspension of these nanoparticles. And those have already been demonstrated. Others have figured out how to do this chemistry. What we were excited about was this initial proof of concept that we can get interfacial activation at the interface in a thermoset matrix incorporated or cured around a rigid inclusion. So this is the image that I've been showing you before, or still from that video. And then this is activation demonstrating that we have mechanophore present at our interface. This is admittedly a very noisy image. I know that there are a ton of hot pixels, and that's because we're trying to see a single layer of molecules, a molecular monolayer, hopefully, of molecules at the interface. So my new PhD student, Anna Freund, is going to spend her dissertation refining this project. So this is very much a work in progress. Stay tuned. I'm really excited about this research direction. All right, so with that, I do want to take a couple minutes and tell you about the cutting tools that we've been developing in my lab that allow us to observe mechanophore activation ahead of a propagating crack tip. So if you'll remember, at the very beginning of this talk, I motivated with this idea of calibration occurs at, or has been performed at, the stress concentration that occurs ahead of a propagating crack tip. In our materials, our silicone specifically, that are too brittle to allow for spontaneous crack propagation, we haven't been able to observe this stress concentration because the crack propagates too quickly through our material. So the solution to this is to start thinking about cutting mechanics. In cutting, you control where the fracture is and where it goes by controlling the force that you're applying with your blade. So this should surprise no one that a sharp blade is better for cutting than a dull blade. You all know this from cutting a tomato or by seeing that surgeons' scalpels are very, very sharp. And all of this relates to the force required to propagate a cut. So what we're interested in is the fracture toughness, or the energy required to create new surface. And for elastomers, what we're going to be cutting, what we're focusing on cutting, is a polymer network that is cross-linked or reacted at different sites and tied together into effectively one single macro molecule, right? So the Lake Thomas theory is a theory that was developed many years ago that effectively said the energy required for cutting is the energy required to break a plane of molecules that your crack has to propagate through in these systems. However, in an elastomer, you have to think about not just the molecules right at the plane or that dotted line where the crack is going to propagate, but there are also a lot of other polymer chains in that network that are performing or have to be moved out of the way and stretched and significantly deformed in order for that crack to be able to move forward. So you end up finding a much larger fracture toughness, or a much larger amount of material participating in the fracture event than was originally proposed. So we're going to be utilizing a modified Lake Thomas theory that predicts a small damage volume. We can't see that, that's little. And then a large deformation zone that I think we can see with our mechanophores. So this green region is what we want to see if we can observe with our cutting experiments. And much of this is going to be drawing on the work out of Shelby Hutchins's group at the University of Illinois Urbana-Champaign, who brought to us the Y-shaped cutting geometry, which is going to allow us to isolate the cut directly over our microscope. So there are a couple benefits, a couple features I want to point out of Y-shaped cutting. Most obviously is you can tell the crack where to go and control how the material fractures. And also, we understand all the mechanics of this. We can measure all of the variables that I'm showing here. So in Y-shaped cutting, we take a sample, we form it into a Y geometry, and then we apply a small preload to the two legs of the cut. And then we're going to shove a razor blade or a blade of some sort into the material. We're going to measure the displacement of our material and the force required to displace the blade into the material. And that's the experiment. From that, we should be able to get a cutting energy, which is going to allow us to then deconvolute the tearing energy or tearing component, if we can get this half angle small enough, from the cutting component. So because Shelby's rig was a little too big to put on a microscope, my graduate student, Tyler Roberts, who's in chemical engineering at Purdue University, developed a new rig. So this was what we drew on the whiteboard, this idea of, instead of displacing the blade into the sample, we could mount the blade on a fixed load cell, fixed in space over a microscope objective, and then move the sample across the blade so that we could watch the cut continuously. So this is what he ended up designing. This is what he built. And you can see here that we have a system of pulleys that are going to allow us to mount the sample. And then this is a $9 load cell and a 3D printed blade holder. And this is what what it looks like installed over a microscope. So this is Tyler's cutting rig. He would be angry if I didn't tell you that this is the soft matter, low friction, Y-shaped cutting experimental rig, or the slicer. I'm sorry, I don't have that on here, Tyler. So this is what this looks like in space. And this is what one of his, these are the parameters that we used for some of these experiments. So we're using the same exact Mechanophore functionalized silicone, still using a 0.6 weight percent loading of the Mechanophore in our system. We get a modulus of roughly 1.4 megapascals for the Young's modulus. We're going to measure millimeter thick samples with a half angle of 20 degrees, and you can see the other properties there. This is what one of these typical cutting curves looks like, where we can pull the F cut directly out of this. We know all our other parameters, so we get a cutting energy of approximately 107 joules per meter squared. Others have already reported the relationship of how sharp a blade is on the energy required to cut, but we did want to make sure that we were reproducing those experiments. So if you plot as a function of blade radius, or how sharp the blade is, there does seem to be a threshold below which there is no relationship between the size of your razor blade or the sharpness of your razor blade and the cutting energy. But as you go to duller and duller blades, there does seem to be some sort of monotonic relationship where the duller the blade, the more energy required to cut. So this is the distinction we're going to make for the rest of this talk, where we're talking about sharp and dull blades. And this is what some of these blades look like. There's a whole science of sharp that you can look at that speaks much, much more explicitly about what is sharp, what is dull, and how to achieve both. But let's get back to mechanophores. So if we look at one of these experiments under our microscope, and now I'm using a different false color image, a different heat map or scale to allow you to really see the concentrations. And you see for this cut material back here, you have this really bright fluorescence response, which is nice, but we only care about the highlighted region. So now we're just going to zoom in to the highlighted region. And what you can see if we rescale our heat map so that we can highlight just the activated portion of mechanophore ahead of our propagating crack tip, we can say that we have approximately 10 microns, maybe a little less, of a large deformation regime that we're able to observe. And if we go through and plot the intensity of this, we can get a little more quantitative with it and actually say how far from our blade we were able to observe fluorescence activation in our material. And so then we went through and looked at the effects of how increasing blade radius changes the size and the shape of the large deformation zone ahead of this propagating crack tip. And these are the fluorescence intensity images of the activated mechanophore as we move from very sharp blades to very dull blades. And you can see there are larger and larger regions of activity as a function of how sharp or how dull the blade gets. And we can quantify this using the normalized intensity mapping. We can go through and say, okay, this is how big or how large of a deformation zone we have as a function of blade radius and get the what we're calling decay distance. And from here you can see a fairly similar relationship. I recognize these are 7 points, but you can see a fairly similar relationship between the sharp and dull blades having no effect and then a monotonic effect on this decay distance as a function of sharp and dull blades. So now that we've done this measurement as a function of stress intensity, we can also map the, oh, did I get the wrong image? No, this is right. So if we take our calibration, our stress to FEA calibration that we have performed and I've talked about ad nauseum, we can then map that and say this is what we think the stresses are quantitatively ahead of the crack tip within our material. And we can confirm this with FEA, where we're showing similar relationships in the increasing size as a function of increasing blade radius or increasing dullness of our blade. So with that, I'm going to wrap up and just say that I hope I've shown you today some of the exciting ways that we can use these mechanophores that have been developed and are actively being developed in engineering modes that allow us to understand more about the deformation behavior of polymer composites, polymer matrices, polymer fracture. And mechanophores have really been a boon to the mechanics of materials, specifically the soft matter materials community. And I'm just very honored to have had the opportunity to share how we have utilized them. Before I wrap up, I do want to acknowledge my collaborators. We have been working closely with Shelby and Chalbeau at UIUC, as well as George Chang, who has provided us all the mechanophores over all of these years, and even Daniel Chang, who came to Purdue University to train us how to synthesize the spiropyran for ourselves. Additionally, I need to thank my research group, the Polymer Interfacial Mechanics Group, both at Purdue and at the University of Delaware, and very recently at the Adhesion Society in New Orleans, where many of the researchers who have participated in this research before and now were able to attend. My funding sources have been have come quite have come from Purdue University as well as the Semiconductor Research Corps, and all of the cutting work has been funded by the Army Research Lab. So with that, I appreciate your attention and I'm excited to take any questions you might have for me.
All
Thank
right,
you.
thank you. Thank you very much for that presentation. We have a few questions from the audience. Start with this. You qualified tests as low, medium, or high adhesion. How do you differentiate these ranges?
Yeah, that's a completely fair question, and I was vague on purpose for those measurements. We have since gone back and performed JKR experiments, which is a Johnson, Kendall, and Roberts contact adhesion test. But for us here, this was simply a proof of concept. So low adhesion is something, if I'm going to quantify it, it's something that occurs where failure between the matrix and the particle is going to occur at single percent strains. Medium adhesion is where we observe the interface stays formed long enough so that you get these jet park debonding events. You store enough energy in your system so that when the interface fails, you observe really large deformations and instabilities within your material. And then for high adhesion, it really came down to, I mean, for these, this was observational, right? But it came down to we never observed interfacial failure. We actually got cohesive failure in our matrix before we observed separation of our interface. And here I kind of glossed through it, but we got even higher adhesion to the point where we started observing the formation of cavities in the material, which is very different from this large scale interfacial separation. So for now, we haven't quantified or we haven't published our quantification of adhesion, and we're just using these as qualitative descriptors.
All right, thank you. All right, next question. Can you back out specific stress tensor components from the Mechanophore illumination? Does the bond scission respond differently to shear versus tension, or is it some global average?
Oh, I love this question. In order to perform our calibration, we needed a scalar quantity, right? We needed a value for a value that we could plot or compare to a value, just the intensity is a scalar quantity, right? Stress is a tensor, totally fair. So we went through a whole series of trying to understand which is the appropriate stress descriptor to utilize in this case. Um, on a single molecule scale, mechanophores, to my understanding, only activate in tension. You have to have a fully extended polymer chain plus a little to pop the mechanophore that we're looking at. So on a molecular scale, the answer is tension and only tension. We are working with nearly incompressible materials. I think the Poisson's ratio is 0.49-ish. So there is going to be a huge amount of activation in our system coming from, even though we're applying a global uniaxial tensile deformation, but we're going to have a huge amount of activation arising from shear that is coming from Poisson effects. And in fact, that's why we see such a bright response right here at the interface of our material. We see this, which can only be a result of really high shear stresses. We have thought deeply about this. Others are thinking deeply about this. We started out with hydrostatic stress. That was not great because we didn't allow any shear to be incorporated into our material. And you can see that it does not have the same shape as what we observe in our real experiments. So to end a really long answer, we settled on max principal stress as our stress scalar that we're using for our magnitudes because we allowed the stress tensor to rotate. And that was the best way we could think of to incorporate the shear stress effects and get something that quantitatively or semi-quantitatively matched really well with what we were observing in our experiments.
Got it. All right, another detailed question here. What is your resolution for the method in terms of strain?
Okay, I could answer that question in several different ways. So the instrument that we're using, the micromechanical load frame that we're utilizing, has strain step resolution that is submicron. Um, so depending on how big of a sample you want, you could have really tiny strains, on the order of hundredths of a percent, right? We are using elastomers. So in our particular case, most of these mechanophores don't even start its exhibiting significant or measurable fluorescence intensity until, I don't know, 40, 50, 60% strain. So we don't want high strain resolution. Um, in terms of measuring the fluorescence activation or the resolution of strain on the microscopic scale with the microscope, that's just dependent on your machine settings. I had a different microscope at my previous institution than I do now, different resolution. It's a function of the camera and the objective you choose.
Got it. Thank you. All right. I have two questions that are related to the implications of this work, and they were not asked by the same person, but I'm going to combine them into a single question and see how you do. Well, then we'll wrap up. All right. So are there any implications of this for first one, a better understanding and prediction of fatigue failure, and
Yeah.
secondly, of synthesis using ball mills? And I realize those are two different things, but I've combined them.
Yes. I am very unfamiliar with synthesis using ball mills, but I'll give it a go. Um, it depends on your mechanophore and the mechanochemistry that you select. I thought I was anticipating the question, and this was not the question. Let's go to
Well, the first one was fatigue failure, so maybe that one is easier.
yeah. Okay, so for fatigue failure, I was watching this video as I was setting up this presentation. Check that the saturation, and so Nasmol is stretching this 3 different times. And to me, it looks brighter the second time than the first time. And it depends on your mechanophore in that if you use like the spiropyran nitro substituted form, those mechanophores tend to stay activated for a couple minutes. If you use something like spirolactam, one of my really early papers showed that that stayed activated in a glassy polymer network for months. If you use spiropyran hydroform or hydrogen form, that stays activated on the order of seconds before it reverts. So if you're looking at sort of damage accumulation under fatigue or cyclic testing or that sort of thing, you have to be really cognizant of the reversion rate of your mechanophore with time and relate that to the strain rate or the application of how quickly are you applying those cycles. Um, I think there is potential there for using this to measure damage accumulation, which I recognize is not exactly fatigue. I think we could do it for fatigue. I don't know anything about synthesis with ball mills. You can design the force of activate, or I've been told that chemists can design mechanophores with very different activation energies or forces required to activate them. So if you want to track how much strain or stress or deformation is being applied to your material as a function of the aggressiveness of your ball mill, you would have to be really intentional about material mechanophore selection and make sure that you got something that was going to be a fair descriptor over the range of mechanical energy that your system is going to be seeing.
All right, I said that was going to be the last one, but I actually got another question twice in different forms, so I'm going to throw this one at you and then we'll wrap up.
Okay.
All right. You used a linear correlation between fluorescence and stress, but the plot did not appear linear. Would a nonlinear correlation improve accuracy?
Completely fair. In the region we care about, I'm going to argue that it is linear. Down here, I also fully agree that it is nonlinear. But what this really is this is our threshold. This is our fluorescence background. our mechanophore hasn't really started turning on here. So I don't think that trying to get really fiddly about catching this part of our curve is going to buy us enough of of an accuracy trade-off to not just treat this as linear. This is before the mechanophore has activated. So we just basically throw this away. And in a lot of our videos or in any of our heat maps, you'll see that we just hand wave and say it's below the activation threshold that we can detect with these particular um, imaging settings. I'm not saying that the mechanophore is not activating before this. I'm saying we can't see it because our detector isn't sensitive enough to it. It's totally fair.
Makes total sense. All right, thank you again for your presentation.
You're welcome. Thank you so much.
Thank you, Professor Davis, for that outstanding presentation. And thank you so much for joining us. Just a reminder that all of the excellent presentations we've had over the last few years as part of the Mechanochemistry discussion series are available to watch anytime on the YouTube channel. We also have a great group of speakers planned for the upcoming months. We hope that you will join us for all of them. Thank you again.